Muhammad Umar Farooq Biological Video. Please make sure to subscribe, like and share. Ask the question for answer and comment about the artwork. You can also question answer to each other for solve the problem. So today we will discuss about the urea excretion from the body. So urea excretion from body is in the form of urea cycle or in the form of another cycle. So overall we will look at the type of the urea excretion. So here I am just drawing the urea cycle, but here we will discuss in the separately. So here is the person which that contain liver and muscle. So the liver and muscle is the important for interchanging the material, a chemical, to excrete the urea by the combination of ammonia. So here I am drawing the muscle which that contain a different reaction. So here muscle contain glutamate. So this glutamate is the glutamic acid which that is produced. So first of all I want to say here is the carbon dioxide and two ammonia molecule will produce a urea. Why? Because 500 ml per gram ammonia is required. It means 500, and here is a 50 ml per gram is water required for urea excretion. On the other hand, if we will talk about the uric acid, which that is more complex compound are present in the bird for excretion, is due to the less amount of water requirement. So the 1 ml per gram uric acid is requirement. So here is the muscle which that is I am drawing, and that's contain a glutamate, and this glutamate will be convert into the glutamine. It means the amine group will be add. So here is the ATP is also used to it to make a ADP, and inorganic phosphate will be separate, not attached. So the glutamate synthetase enzyme used to it to make a glutamine. So this glutamine go into the muscle liver. So here I am drawing the liver, which does contain a glutamine, which does come from the muscle, and that is active by the enzyme, and water will be absorbed to produce ammonia, and this ammonia will be two molecule of ammonia convert into the urea. So this urea is the production, and here is the hydrogen bicarbonate which that's convert into the HCO3 hydrogen carbonate ion, and acidic medium will be created for decrease the acidity when. So here is the carbon dioxide will come from the carbonic acid to attach into the ammonia to produce urea. So this urea will be produced. So let's go, we will talk about this carbon dioxide will come from the carbonic acid and splitting, and here is if not produce urea, so the glutamate will be converted into the alpha ketoglutarate by the action by the action of alanine. So alanine will convert into the pyruvate by the action of alpha ketoglutarate. So these I am drawing here again the muscle. When that's back the muscle, here is the pyruvate will be gluconeogenesis to produce glucose. It's the reverse mechanism, and this gluconeogenesis to produce basically this glucose will go into the muscle cell. So here is the gluconeogenesis can be occur into the liver, and that glucose will be go into the muscle. So this is the muscle which that's convert again pyruvate by the glycolysis, and this pyruvate is convert again into the alanine, which that is same like a liver into the liver, but here is the alanine which that is bind by the glutarate to convert alpha ketoglutarate is same like a liver. So here is the alanine aminotransferase enzyme used to produce alanine from pyruvate in the presence of glutarate. So that's go into the alanine into the again back into the liver, and that's again will be pyruvate produced. So this is known as glucose alanine cycle. It means the glucose produced to again produce alanine, and that's go into the liver. So here is the alpha ketoglutarate will be produced by the glutarate reaction. If alanine will react to the glutarate to produce a alpha ketoglutarate, and in the result of amino group amino acid will be bind to the glutarate to the alpha ketoglutarate to basically produce, or amino acid will convert into the valine isoleucine succinyl CoA conversion, and that's go into the glycolysis again. So this R reaction is due to the urea production. So here is the urea will be produced, and that's go into the bloodstream from the liver. So that is go into the glomerulus, and that's after the filtration into the glomerular corpuscle or Bowman capsule to excrete the urea into the urinary system from the kidney. So here I am drawing the glomerulus, and that's all thing is done. So here I will draw basically mitochondria, which that is the we will look at the urea cycle to understand. This reaction is not a urea cycle. This reaction is also present in the liver, but this is the cycle is glucose alanine cycle, which that are also produce the urea by basically by a glutamate to glutamine production, glutamine to glutamate production. But here is the glutamine will be converted into the ammonia to produce a urea. So here is the mitochondria, which that's contain a carbamoyl phosphate. Carbamoyl phosphate, it means the phosphate group is attached with the carbamyl. So that's the basically here is the O trans carbamylase OTC will be bind to produce here is the from outer side cytosol to enter the transporter of ornithine to produce both combination to produce citrulline, and this citrulline transporter will be excrete from the mitochondria to citrulline is here, and that use the ATP to convert and that is that phosphate group is attached it, and this produce citrulline AMP, adenosine monophosphate. So AMP is the citrulline AMP is the structure is look like this. So here is the arginosuccinate synthetase enzyme used to produce arginosuccinate in the presence of aspartate, and that's aspartate will be detach the AMP, and that's AMP will be detached from it, and that's arginosuccinate formula is looking like this. And that's the other is the here is the compound will be separate, and that is arginosuccinate lyase enzyme to release that basically carboxylic 2 carboxylic group is looking, that is the bicarboxylic acid carboxylic ion which they are separate. So here is the Rg9 will be produced in the presence of arginosuccinate lyase to Rg9 production. So here the water will be enter in here, and that's produce a nitrogen to produce here is the water will be go into the here is the H hydrogen ion will move in here, and that's here is the hydrogen ion will move here, and this will be produce basically urea. So the urea structure is here. So these will be separate is like that if I draw again this structure, so here the water will be add, and addition of water will be release the urea. So this all compound will be formed for the evolutionary process to decrease the level of water for excretion of ammonia in the water body animal. So here's the citrulline transporter again go inside to attach the carbamoyl phosphate to produce citrulline. So basically ornithine will be add. So the ornithine will be add here. So that's excretion will be occur. So that's all thing is done here. So thanks for watching. Please subscribe my channel and watch another video.